Essentially, all cells carry out glycolysis. There are 10 reactions in glycolysis, same in all of the cells, except the rates differ. Glycolysis breaks down into two phases. The first phase converts glucose to 2-glycerolaldehyde 3-phosphate. The second phase produces 2 pyruvates. The products of glycolysis are pyruvate, ATP, and NADH. There are 3 possible states for pyruvate. In aerobic conditions, pyruvate is oxidatively decarboxylated to produce acetyl-CoA, which then enters the TCA cycle. Or in anaerobic conditions, pyruvate is reduced to lactate in oxygen-starved muscle or into ethanol in yeast. The first reaction of glycolysis is the phosphorylation of glucose. The first reaction is a primary reaction, which means it has a large negative delta G. In this reaction, ATP is used, which makes phosphorylation of glucose spontaneous. The enzyme used are hexokinase or glucokinase, and there is a difference between these two enzymes. Hexokinase and glucokinase act to phosphorylate glucose and keep it in the cell. The Km for hexokinase is 0.1 millimolar. Km means the binding constant to glucose, and glucokinase Km is 10 millimolar. The cell normally has about 4 millimolar blood glucose concentration. So, with this said, hexokinase is the one normally active. Hexokinase is the one normally active, while glucokinase is only active when glucose is rich in the blood. So here's the reaction. Glucose using ATP, hexokinase or glucokinase. Remember again, these enzymes depend on the blood glucose concentration. And then with that it goes to glucose 6-phosphate. The negative charge on this makes it stay into the cytoplasm. So glucose goes from the extracellular fluid into the cytoplasm, and it cannot come back due to the negative charge. The second reaction of glycolysis is called the phosphoglucoisomerase reaction. In this second reaction, glucose 6-phosphate goes to fructose 6-phosphate using PGI, phosphoglucoisomerase. This reaction involves an opening of the pyridinose ring, proton extraction leading to the enodiol formation, and the proton addition to the double bond followed by a ring closure. The third reaction is the phosphofructokinase reaction. This reaction is the committing step with a large negative delta G, which is highly regulated. ATP inhibits the reaction, while AMP is the reverse inhibition. This reaction goes from fructose 6-phosphate to fructose 1,6-bisphosphate with the addition of magnesium. Again, PFK is phosphofructokinase. Again, the name of the third reaction is the same as the enzyme. You add ATP and magnesium 2+ in order for this reaction to go through. The fourth reaction of glycolysis is called aldolase reaction. This reaction makes 2 products, DAP and glycerolaldehyde 3-phosphate. Animal aldolases are class 1 aldolases. Class 1 aldolase form a covalent shift-based intermediate between the substrate and the active site lysine. The enzyme used in this reaction is fructose biphosphate aldolase. So in this reaction, again, you form 2 products. You go from fructose 1,6-bisphosphate with the enzyme fructose biphosphate aldolase. You create DAP and glycerolaldehyde 3-phosphate. The last reaction of the first phase is triphosphate isomerase. The goal of this reaction is to convert that obtained previously from the previous reaction to glyceraldehyde 3-phosphate. This reaction involves an enediol mechanism. Active site glutamine acts as general base. Again, the enzyme takes the name of the reaction, which is triphosphate isomerase, also known as TPI. So again, this reaction goes from that to glyceraldehyde 3-phosphate. But remember, it forms an enediol intermediate, which is this right here. And then it goes to the glyceraldehyde 3-phosphate. Again, this is the end of the first phase, so you got one glyceraldehyde 3-phosphate in the fourth reaction, and now you have the second glyceraldehyde 3-phosphate needed to proceed on. Now we're in the second phase. Remember, the metabolic energy produces 4 ATP, but the net yield for glycolysis is 2 ATP. Remember, it uses 2 ATPs in the process. That's why you end up only with 2 ATP at the very end. The first reaction of the second phase and the sixth reaction overall is called the glyceraldehyde 3 dehydrogenase reaction. In this reaction, glyceraldehyde 3-phosphates oxidize to yield 1,3-bisphosphoglycerate. The energy yield from converting an aldehyde to a carboxylic acid is used to make 1,3-bisphosphoglycerate and NADH. This reaction has mechanism involves a covalent catalysis and a nicotinamide coenzyme. So this reaction again goes from glyceraldehyde 3-phosphate using the enzyme glyceraldehyde 3 dehydrogenase to make 1,3-bisphosphoglycerate. Again, this reaction of an enzyme sulfohydryl with the carbonyl carbon of a glyceraldehyde 3-phosphate forms a thiohemiacetal, which loses a hydride to the NADH plus to become a thioester. Phosphorylasis of this thioester releases 1,3-bisphosphoglycerate. So it releases this in this reaction. And that's how you get that. The seventh reaction is called the phosphoglycerate kinase. In this reaction, ATP is created. This is one of the 2 ATPs created in all of glycolysis. This reaction, you go from 1,3-bisphosphoglycerate to 3-phosphoglycerate. Again, the enzyme in this reaction is the same as the name, so phosphoglycerate kinase. It uses magnesium 2+ and ADP, and it goes from 1,3-bisphosphoglycerate to 3-phosphoglycerate, and it creates your ATP. So that's one of the 2 ATPs created in this second half of glycolysis. The eighth reaction of glycolysis is the phosphoglycerate mutase reaction. This enzyme repositions the phosphate from 3-PD phosphoglycerate to 2-PD, which is 2-phosphoglycerate. So all it does is essentially move the phosphoryl group from the third carbon to the second carbon. Again, the enzyme this reaction uses is the same name as the name of the reaction, which is phosphoglycerate mutase. I abbreviated to PGM. So again, it takes 3-phosphoglycerate to 2-phosphoglycerate. So all it's doing, it's moving this phospho group all the way over here to the second carbon, as you can see it over here. Also note that in this reaction there is a phosphohistidine intermediate. The ninth reaction is the enolase reaction. The overall delta G of this reaction is very low. It's actually 1.8 kilojoules per mole. How can it create PEP with such low energy? This reaction is actually energy content. What happens here, enolase just rearranges the two phosphoglycerate to a form from which more energy can be released in hydrolysis. Again, this goes from 2-phosphoglycerate to PEP. With the help of magnesium 2+, 2-phosphate glycerate gets converted to phosphoenolpyruvate, PEP. And that's what PEP means, is phosphoenolpyruvate. And last but not least is the 10th reaction of glycolysis, which is pyruvate kinase. This reaction has a large negative delta G, which is for regulation. So you have 3 reactions that regulate glycolysis, which is the first reaction, the third reaction, and this last reaction. This reaction is allosterically activated by AMP and fructose 1,6-bisphosphate. This reaction is allosterically inhibited by ATP and acetyl-CoA. So here's the last reaction. You go from PEP plus hydrogen plus ADP with magnesium 2+ and potassium gives you pyruvate and your last ATP. So in total you should have 2 pyruvates and 2 ATPs that you made. I hope the covering of this 10 reaction of glycolysis was helpful, and till next time.